Hello, my name is Kewalin Samart. I am a recent graduate of Michigan State University. I have been working with Amy Tonioli, Dr. Jen Krishnan, and Dr. Janani Ravi on integrating multiple transcriptome-based methods to repurpose drugs for infectious diseases. Infectious diseases cause millions deaths worldwide, leading the overuse of antibiotics, causing the rise of antibiotic resistance in pathogens. Host-directed therapies combined with computational drug repurposing can be a great promise for drug discovery against antibiotic resistance. And with the fact that efficacious drug will successfully reverse the disease signature with minimal side effects. And over the past 15 years, many transcriptome-based methods have been developed towards finding disease-drug reversal relationship. Although we have many connectivity scores proposed by different studies, they all differ from each other in their choice of specific similarity metric and how they are combined, normalized, and background corrected. Therefore, the goal of our project is to integrate different transcriptome-based disease drug connectivity score to repurpose drug for infectious diseases, starting with tuberculosis or TB. And the reason why we chose to study TB first is that we have a bunch of public available data in the database. And TB is also a critical infectious disease. Our workflow starts with downloading raw TB expression data from GEO, and then we perform differential gene expression analysis to construct 26 active up and down TB signatures. We only use human samples and 978 landmark genes with significant genes cut off at adjusted p-value less than 0.05. We divided our workflow into two approaches. The first approach, we used 26 individual TB signatures, and the second approach, we aggregate the 26 individual TB signatures into just one aggregated signatures using weighted average method. Then we use the TB signatures as query to compare similarity with FDA-approved drug signatures in the LINKS database, and we prioritize the potential drugs candidates by the 6 score, which are CS score from CMAP 1.0, WCS, NCS, and Tau score from CMAP 2.0, X score and XP from correlation-based method. And then for individual signatures, we will get 26 drug lists for a given method, and then we select top 20 to 25 drugs that partially or fully reverse at least 50% of the 26 TB signatures. And then for both approaches, we will subset final drugs that come up in at least 2 out of the 3 methods, CMAP 1.0, 2.0, and correlation-based method. As a result, we finally identify 20 potential drugs from individual signatures and 16 potential drugs from aggregated signatures. And then we evaluate the result by performing literature evidence search. And it turns out that we have 12 out of 20 potential drugs and 5 out of 16 potential drugs from individual and aggregated signatures, respectively, with literature evidence against TB infection. Furthermore, we identify and validate top mechanism of action of the top drugs using feature exact test. And then we compare the MOA result back to the enrichment analysis result, which are the enriched TB pathway from aggregated signatures and individual TB signatures. We also evaluate the aggregated signature by looking at the number of shared enriched GO pathway across up and down individual and aggregated TB signatures, as you can see in figure number 2. And here on the x-axis, we have overlapping set, and then on the y-axis, we have the intersection size, which indicates the number of shared enriched GO pathway across different sets of signatures. And as you can see that we have total 45 shared GO pathway across up and down individual and aggregated TB signatures out of about 1,000 out of about 1,000 pathways. In conclusion, our integrated workflow has successfully addressed heterogeneous datasets and identified efficacious HDT with MOAs and enriched pathway for active TB infection. And based on literature evaluation, we observed that predicted drug candidates from individual TB signatures are more promising compared to the aggregated signatures. In the future, we will perform baseline comparison to identify appropriate cell lines for further experimental validation and improvement of the workflow. Then we will extend the workflow to repurpose drug for other critical infectious diseases. Also, we will implement the drug repurposing pipeline in an R package. I would like to thank all the funding and would like to acknowledge everyone in the Jerari and the Krishna Lab, also ISMB 2022 for the opportunity to present my work today. And if you have any question, please feel free to contact me. Thank you very much for your attention.